You know, usually when we look back at the history of a major scientific breakthrough, there's this expectation of a neat, tidy timeline.
Oh, absolutely. We love the myth of the straight line. It's, incredibly comforting to think that scientific progress is entirely predictable and clean.
Right, like a genius in a pristine lab has a sudden epiphany, they run a perfect experiment, and boom, humanity is saved.
Yeah, but then you step into the world of oncology and drug development, and suddenly that straight line looks more like, well, a tangled ball of yarn that a cat has been playing with for a decade.
Exactly. We are looking at a landscape today that it's messy, it's full of sheer accidents, blunders, and honestly, some profoundly human moments.
It is the absolute definition of a scientific roller coaster.
So today, we're taking you, the listener, on a deep dive into that messy, accidental history of cancer drug discovery. We're drawing entirely from this incredible book by Jie Jack Li called Cancer Drug Discovery.
Yeah, and it's a fascinating read because understanding the bizarre winding path of how we got these drugs really helps us appreciate just how complex our own biology actually is.
Our mission today for you is simple. We're going to bypass the heavy medical jargon. Instead, we're going to uncover the aha moments, the dumb luck, and the dogged persistence that led to the medical marvels we rely on today.
Which is, frankly, the most interesting part of the science anyway.
Okay, let's unpack this. To cure cancer, humanity first had to figure out what was actually causing it. And to do that, we have to travel back to 18th century London.
Right, the 1770s. The Industrial Revolution is just taking off, and coal is rapidly becoming the major heating fuel in London.
Which meant the city was essentially running on fire.
Exactly. And chimneys were constantly getting clogged with this heavy, thick coal soot.
So homeowners needed a way to clean those really narrow, twisting flues. And they employed young boys, sometimes as young as 5 or 6 years old, as chimney sweeps.
Because these kids were small enough to agilely climb down the brickwork and literally scrape away the blockages.
It was a brutal, brutal job. And in 1775, a London surgeon named Percival Pott noticed something horrifying.
Yeah, he saw a massive spike in a very specific rare form of scrotal cancer among these young chimney sweeps right as they hit puberty.
But Pott noticed this really fascinating comparative detail. He realized that French chimney sweeps, who were doing the exact same grueling work, weren't getting this cancer nearly as much.
Right, and he dug into the lifestyle differences and found the answer.
Which was so simple. The French sweeps bathed daily. The London sweeps rarely washed.
So Pott connected the dots. The culprit was the coal soot getting physically lodged in the furrowed folds of the skin over long periods of time.
Wow.
And this observation was monumental because it birthed the chemical carcinogen theory, the groundbreaking idea that a substance in the external environment could directly cause cancer through prolonged contact.
But, establishing a theory and actually proving it in a lab are two very different things.
Oh, completely. For over 100 years, scientists across Europe tried to recreate Pott's findings in animals to definitively prove that chemicals cause cancer.
And everyone failed. Until 1915, when a Japanese scientist named Katsusaburo Yamakiwa finally cracked it.
Yeah, and his methodology is just a testament to absolute obsession.
To put it mildly, he and his assistant took rabbits, and they painstakingly painted crude coal tar onto the inner surfaces of the rabbits' ears.
3 times a day, every single day.
After 250 days, I try to wrap my head around that level of endurance. That is literally watching paint dry. But for nearly a year, smelling crude tar, repeating the same monotonous motion.
It's wild.
How do you not just throw your hands up and quit by day 150? Most scientists before him simply gave up because the tumors took way too long to form.
Well, it requires a profound, unwavering belief in your hypothesis. Yamagiwa knew the biological timeline might be slower than human patience.
Right.
And his persistence paid off. Out of 137 rabbits, a few finally developed grotesque skin tumors.
And Yamagiwa was so overjoyed by this grim result that he actually wrote a haiku. I love this detail so much. He wrote, "Cancer was produced. Proudly I walk a few steps."
Yeah, it's dark, but you can really feel his absolute triumph there.
Oh, totally.
But what's fascinating here is that while Yamagiwa proved that soot and coal tar cause cancer, the scientific community was still largely in the dark about the mechanism.
Right.
We knew that these chemicals caused tumors, but we didn't know what the chemical was actually doing to the body at a microscopic level.
Wait, hold on. If we knew soot caused tumors, how did we get from soot is bad for the skin to knowing what it physically does inside the cells? It feels like there's a massive missing link there between observing a tumor and understanding the biology.
Oh, there was. That missing link didn't arrive until the 1970s, thanks to a scientist named Bruce Ames.
Okay.
Ames realized that we needed a drastically faster way to test chemicals. You can't paint rabbit ears for 250 days every time you want to evaluate a new industrial compound.
No, obviously not.
So, Ames created a brilliant, rapid assay using Salmonella bacteria.
Bacteria that reproduce a new generation every 20 minutes.
Exactly the point. The elegance of the Ames test is in its design. He took a strain of Salmonella that lacked the ability to synthesize a specific, necessary amino acid.
So without it, they couldn't grow.
Right. Ames exposed these crippled bacteria to various suspected chemical carcinogens. If the chemical caused the bacteria's DNA to mutate in a specific way, that suddenly allowed them to produce that amino acid.
Oh, the bacteria would thrive and multiply.
Exactly. It was a clear, visual, positive test.
So if the bacteria suddenly start growing, the chemical fundamentally mutated their genetic code.
Yes. Ames used this to prove a massive concept. Carcinogens are actually mutagens. They physically damage and alter the DNA code itself.
So cancer isn't just an irritation of the skin, it's a literal rewriting of the cellular instruction manual.
Arriving at that truth, that cancer is ultimately a disease of our genes, was not a straight path. Science got it spectacularly wrong before getting it right.
Oh, we definitely have to talk about the great blunders. I mean, the 1926 Nobel Prize in Medicine is the perfect example of this messy timeline.
It really is. It was awarded to a Danish pathologist named Johannes Fibiger.
And he claimed he had found the cause of cancer, once and for all.
Right. He was studying wild rats in a Copenhagen sugar refinery and noticed massive stomach growths. Upon dissection, he found that the rats had eaten cockroaches infected with a specific roundworm.
And Fibiger confidently claimed these parasitic worms caused stomach cancer.
He did. And the Nobel Committee absolutely loved it. They awarded him the highest prize in science.
But you have to wonder why they were so eager to believe him. It's because an infectious worm fit perfectly into the medical paradigm of the early 20th century.
They knew how to fight infections.
Exactly. They desperately wanted cancer to be an external enemy they could just kill. But years later, nobody else could replicate Fibiger's results.
Right. It turned out the cancer wasn't cancer at all. It was just abnormal tissue growth caused by a severe vitamin A deficiency in the rat's diet.
A literal Nobel Prize for a vitamin deficiency.
It's a stark reminder that even the most prestigious institutions can be blinded by compelling data if it tells them a story they want to hear.
Right. The true origin of cancer wasn't an external worm or a foreign invader. The terrifying truth lay hidden inside our own normal DNA.
Which brings us back to the 1970s and a discovery by two scientists, Michael Bishop and Harold Varmus.
Because up until this point, a lot of researchers still thought viruses were the main culprits, essentially injecting foreign cancer-causing genes into our cells.
Exactly. So Bishop and Varmus used specialized DNA probes to search for these supposed foreign cancer genes.
And what they found completely flipped our understanding of biology on its head.
It did. These genes, which they called proto-oncogenes, are actually normal, essential genes that already exist inside every single one of our cells.
Wow.
They have been there throughout billions of years of vertebrate evolution.
So they're entirely normal genes that govern cell growth, but they just have the potential to go rogue and cause cancer if they mutate. We are essentially carrying the seeds of our own destruction.
Unfortunately, yes. And this realization was complemented by another monumental concept, Alfred Knudson's two-hit hypothesis.
Right. Knudsen was studying retinoblastoma, a rare childhood eye cancer.
Yes. Through brilliant statistical analysis, he realized that getting this cancer required two specific genetic mutations.
So if you inherit one broken gene, that's the first hit.
Right. But the cancer doesn't develop until a second random mutation breaks the backup copy. That's the second hit.
And this led to the discovery of tumor suppressor genes.
Exactly. If we look at the mechanics of a cell, it's a highly regulated, high-performance machine. Proto-oncogenes, when they mutate into oncogenes, act like a jammed gas pedal.
They tell the cell to divide relentlessly, foot to the floor.
But the cell also has brakes. The tumor suppressor genes. Their job is to monitor the system, stop division, and repair damaged DNA.
But if those suppressor genes mutate and lose their function, if you get that second hit, it's like somebody literally cutting the brake lines on a speeding car.
Yes. And if we connect this to the bigger picture, this exact realization is what finally gave scientists a clear biological target.
Okay.
Once we understood that cancer was driven by rogue DNA replication and runaway cell division, the entire field of oncology shifted toward finding ways to target and stop that specific mechanical failure.
So by the 1970s, we've mapped the enemy's blueprint. We know cancer is a runaway replication machine built out of our own mutated DNA.
Right.
But that creates a terrifying problem.
Yeah.
How do you throw a wrench into a machine that is built from the exact same materials as the rest of your body?
It's incredibly difficult. And the first answer to that didn't come from a pristine lab, but from the battlefield.
Yeah, it is one of the most paradoxical stories in modern medicine. During World War II, an American merchant ship called the John Harvey was secretly carrying a massive payload of mustard gas.
And it was bombed by the Luftwaffe in the harbor of Bari, Italy. The gas mixed with the oil and water, exposing hundreds of sailors.
An Army medical officer, Dr. Stuart Alexander, was investigating the casualties and noticed a very peculiar pattern in the blood work of the victims.
The mustard gas had completely wiped out their white blood cells.
Because white blood cells divide very rapidly, just like cancer cells do.
Exactly. And this grim observation made its way to two scientists at Yale, Alfred Gilman and Louis Goodman. They reasoned that if mustard gas effectively destroys fast-dividing white blood cells, maybe a scientifically tweaked version could destroy fast-dividing cancer cells.
So they synthesized a liquid version, nitrogen mustards. They administered it to a patient with advanced lymphoma, and the tumors practically melted away.
It was the birth of chemotherapy.
It's just wild that it came from chemical warfare.
It was a crude weapon initially, but it proved that you could use systemic chemicals to fight cancer. However, creating these DNA-targeting drugs is a game of molecular tightrope walking.
Because you want to kill the cancer, but not the patient.
Exactly. Which brings us to the incredible story of temozolomide. In the 1980s, Malcolm Stevens and his lab created a molecule called midazolamide.
Right.
It was designed to deliver a nitrogen mustard-like warhead directly to cancer cells. In mice, it was an absolute magic bullet. It wiped out tumors with a single dose.
But when they took it to human trials, it was a profound disaster. It destroyed the patient's blood platelets.
The drug was effectively dead. A competitor even jokingly labeled the doomed venture as only last one.
Ouch.
But we have to look at why it failed. That drug had a bulky chlorethyl group attached to it. That bulky group was too aggressive. It was indiscriminately smashing into healthy bone marrow, causing catastrophic damage to the platelets before it even reached the tumor.
But Stevens' lab had one other related compound left on the shelf, temozolomide.
Right.
It was almost identical to the failed drug, but they swapped out that bulky, aggressive chloroethyl group for a sleek, simple methyl group.
And that single structural substitution saved the drug. The methyl group made the molecule incredibly stable in the bloodstream.
Like a Trojan horse.
Exactly. Floating harmlessly through the body until it crossed the blood-brain barrier and hit the slightly alkaline environment of a brain tumor. Only then would it snap open and release its payload.
It became a billion-dollar blockbuster drug. Because of its ability to penetrate the blood-brain barrier, it effectively treats gliomas, a deadly brain tumor.
It literally allowed bed-bound terminal patients to get up, go about their normal lives, and even go on vacation.
Doctors called it the Lazarus phenomenon. Here's where it gets really interesting. Can you explain what these alkylating agents are actually doing when they finally open up inside the cancer cell?
Yeah, so when temozolomide activates, it releases a highly reactive agent that covalently bonds to the DNA bases inside the cancer cell.
Okay.
The DNA molecule is a double helix, and in order for a cell to divide and multiply, that double helix has to unzip into two separate strands so it can be copied.
So if I'm picturing this, it's like supergluing a zipper shut. The drug comes in and chemically glues the two strands of the DNA double helix together. The cancer cell tries to pull the zipper down to copy itself, realizes it's completely stuck, triggers an alarm, and ultimately self-destructs.
That is a perfect analogy. It chemically locks the zipper, but we have to acknowledge the collateral damage here.
Right.
While these drugs are highly effective, they are fundamentally indiscriminate. They attack any fast-dividing cell. That includes your hair follicles, the lining of your stomach, and your immune cells.
Which explains why the side effects of early chemotherapy, hair loss, severe nausea, immune suppression are so brutal.
Exactly.
Now, we've talked about drugs born from the tragedy of poison gas, or meticulously tweaked by chemists. But other major chemotherapy drugs were discovered by pure, unadulterated accident.
Oh, absolutely. Let's look at cisplatin. This is a drug that completely revolutionized the treatment of testicular cancer.
Turning a largely fatal diagnosis into a disease with a cure rate of over 90%, famously saving the cyclist Lance Armstrong.
Right, and its discovery was a complete serendipitous fluke.
In 1965, a physics professor named Barney Rosenberg wasn't even looking for a cancer drug.
No, he was trying to see how electric currents affected bacterial growth. He put platinum electrodes into a bacterial bath, assuming platinum was a totally inert metal that wouldn't chemically react with anything.
And he was completely wrong.
The bacteria didn't just stop dividing, they started growing endlessly, stretching out into massive strands 300 times their normal length.
They look like massive piles of spaghetti under the microscope.
And it wasn't the electricity causing the spaghetti effect at all.
Right, Rosenberg eventually realized the platinum electrodes had slowly dissolved and chemically reacted with the ammonium chloride buffer in the bath, creating an entirely new compound, cisplatin.
Which is just wild.
But why did it make them turn into giant spaghetti strands?
Well, cisplatin acts similarly to the superglue we just talked about. It had chemically paralyzed the bacteria's ability to copy their DNA and divide. But, and this is the fascinating part, it didn't stop their cellular growth machinery. So the bacteria couldn't split in two, but they kept physically growing, elongating into these massive strings.
So Rosenberg realized that if this accidental platinum compound could stop bacteria from dividing, maybe it could stop cancer cells from dividing.
Yes, serendipity at its absolute finest.
But sometimes humans don't even need to make the accidents. Sometimes nature has already done the complex chemistry for us, hiding in the most extreme environments on the planet.
Oh, this is one of my favorite stories. In 1969, the National Cancer Institute was running a massive screening program testing marine life for potential medicines.
And they found incredible anti-tumor properties in a simple Caribbean sea squirt, a little gelatinous marine animal called a tunicate.
The drug hidden inside it is called trabectedin. It is insanely potent, but the timeline here is wild. It took 46 years to get this drug from discovery to the market.
Wait, hold on. 46 years.
Yeah, the delay was purely a supply problem. The sea squirt produces such a microscopic amount of the active compound. It literally took 1,000 kilograms of sea squirts to extract just 1 single gram of the drug.
Okay, if they identified this incredible anti-cancer molecule in a sea squirt in 1969, why are they spending decades trying to dredge thousands of pounds of marine animals out of the ocean? Why not just hand the chemical structure to a chemist and say, mix this up in a beaker on day one?
Because nature is infinitely more sophisticated than our best laboratories.
Fair enough.
The structural complexity of these marine molecules is baffling. They have intricate 3D shapes, specific chiral centers, and delicate bonds that are incredibly difficult to replicate artificially.
Okay, so they couldn't just build it.
Right. It took decades of trial and error before a Harvard chemist, a Nobel laureate named E.J. Corey, finally figured out the exact sequence of synthetic steps required to build trabectedin from scratch in a lab.
And then, after all that effort, the ultimate plot twist happened.
It really is. In 2015, long after the drug was finally approved and saving lives, researchers made a stunning realization through advanced genetic sequencing.
The sea skirt wasn't even producing the drug.
No. It's almost comical. A tiny symbiotic bacterium living inside the sea squirt was the actual manufacturer of the trabectedin all along.
We spent decades focusing on the animal, completely missing the microscopic factory living inside it.
Exactly.
Okay, so we've covered supergluing the DNA zipper shut, whether with mustard gas derivatives or accidental platinum compounds. It works, but as we said, it's highly toxic.
You're essentially nuking the body to save it.
Right, but what if we didn't have to kill the cancer cells at all? What if we could just talk them down off the ledge?
This brings us to the realm of epigenetics, and it represents a massive paradigm shift in how we approach oncology.
Because we spent 50 years trying to poison cancer.
Exactly. But the story of epigenetics suggests we can just convince the cells to behave normally again. It starts in 1966 with a scientist named Charlotte Friend.
She was studying mouse leukemia cells in a lab. One day, she accidentally left these colorless malignant cancer cells sitting in a harsh chemical solvent called DMSO.
And when she came back and checked the tubes, the cells had turned a distinct pink and red.
Because they had started producing hemoglobin.
Exactly. The leukemia cells had basically stopped being cancer and had reverted to acting like healthy, functioning red blood cells.
She literally ran down the hall showing everyone the test tube. It was visual proof that a cancer cell's fate wasn't permanently sealed.
But there was a major hurdle. DMSO is an industrial solvent. It's way too toxic to pump into human patients.
So two researchers, Paul Marks and Ronald Breslow, dedicated years to creating a safe, non-toxic pharmaceutical version of this effect.
Eventually, they synthesized a compound called SAHA, also known as varinostat.
But when they took this elegant compound to big pharma executives, they were literally laughed out of the room.
Yeah, the executives looked at the chemical structure and thought it would be far too toxic to ever work in a human.
So Marks and Breslow did what any self-respecting stubborn scientist would do. They started their own biotech company, proved the executives wrong, ran successful trials, and ended up selling the drug to Merck for a fortune.
Which is amazing. But this raises an important question. How does a cell change its behavior so drastically without actually changing its underlying DNA sequence?
Right, because SIDA doesn't mutate the DNA. The code itself hasn't been altered.
Exactly. How do you cure a cell without killing it or altering its genetic code? To understand epigenetics, you have to look inside the nucleus.
Okay.
Your DNA doesn't just float around loosely like a bowl of spaghetti. That would be chaotic. Instead, the DNA wraps tightly around specific proteins called histones, very much like thread wrapped around a wooden spool.
Okay, I'm with you.
Epigenetic drugs, like SA, which belong to a class called HDAC inhibitors, don't change the genetic code itself. Instead, they change how tightly the DNA is wound around that spool.
Oh, I see.
They dictate which parts of the genetic code the cell is actually allowed to read.
Let me try to visualize this. Imagine the cell's DNA is a giant, comprehensive instruction manual for how to build and operate every part of the body.
Sure.
Cancer hasn't gone in with an eraser and physically rewritten the words in the manual. Instead, cancer has basically taken a bunch of paper clips and clamped the wrong pages together, so the cell simply cannot read the instructions that tell it to stop dividing and mature into a normal cell.
That is a brilliant way to visualize it. The vital genetic information, the blueprint for healthy behavior, is still entirely intact. It's just hidden from the cell's reading machinery.
So these epigenetic drugs like SAHA, they act like somebody walking into the library, picking up the manual, and simply removing the paper clips.
Yes.
Suddenly the cell can open those pages, read the correct instructions again, and remember how to be a healthy cell. It stops dividing relentlessly and starts producing hemoglobin.
It's teaching an old mutated cell new tricks,
Good.
or rather removing the barriers so it remembers its old tricks.
So what does this all mean? We have gone from young chimney sweeps in 18th century London, mapping the origins of the disease.
Right.
We've witnessed the horrors of mustard gas in World War II, birthing the first chemotherapy. We've seen platinum electrodes acting weirdly in a physics lab, and a microscopic bacterium living inside a Caribbean sea squirt providing a miracle drug.
All the way to the cutting-edge science of removing molecular paperclips through epigenetics.
It is a dizzying, spectacular trajectory.
It really is.
And for you listening, I think it highlights that human progress against our oldest, deadliest foe isn't a neat, tidy timeline pointing straight up. It is a messy, deeply human story.
It requires scientists collaborating across decades, stubbornly questioning established dogma, and relying on a very healthy dose of serendipity.
It absolutely does.
And there's one final thought I want to leave you with today, especially regarding that sea squirt story. It is a profound realization. If life-saving billion-dollar chemotherapy drugs are hiding inside microscopic symbiotic bacteria within rare marine animals, we have to ask ourselves a difficult question.
What's that?
How many future cures are we losing every single day due to the destruction of our oceans and the loss of global biodiversity? It is a stark reminder that protecting the planet might literally be the key to saving ourselves.
That is a really powerful point to end on. We started this deep dive talking about how we expect scientific progress to be a straight, clean line, but the reality is a tangled, messy web. And as we've learned today, that messy web is exactly where the miracles are hiding.